interesting picture I've ever taken from my study. So I have a lab set up in the basement of the building, and one day I found a cockroach that was residing in it. It was kind of creepy, but I'm glad that my study has become a really nice niche for wildlife in the lab. Minnesota is known as the land of lakes. And when it comes to the summer season, we kind of become the land of algae bloom. You can see patches of algae that are on the surface of all the water bodies. And they just grow there, and they start killing fish and degrade the water quality and making all the recreational activities undesirable. The reason why Midwestern Cornbelt is such a critical region is because we do not just cause the regional or statewide problem. We also cause problem up and down. So to the south, every year we deliver tons of nitrate to the Gulf of Mexico, which causes annual eutrophication problems. And I'm sure that's all over the news. And to the north, we also deliver excess phosphorus to Canada from the Red River, and they're kind of blaming us for it too. So because of this issue, in order to address the issue, Minnesota Pollution Agency, known as MPCA, has proposed a nutrient reduction strategy for Minnesota last year. The draft was available online. Oh, I got it here. And in that strategy, there are actually two maps here. So to save time, I'm actually just going to draw it. So forgive me if my drawing is really terrible. So if you imagine, this is the state of Minnesota, and a lot of issues occurs in south central Minnesota, so over here. It is both a critical region for nitrate pollution and phosphorus pollution. So a lot of studies have occurred over in that region, same as our study. Our research field site location is actually quite close to Iowa border. It's Iowa down here. So it's over here in south central Minnesota. Unfortunately, we were supposed to have field set up ready last year, but due to a little miscommunication with the contractors, instead of digging a trench, they kind of created a lake, because our field site was adjacent to the stream, and they dug the trench too deep, so the water keeps feeding back into the trench. So that's why I switched my study to a lab scale. So the idea, well, there are a couple of best management practice known as BMPs in the Midwestern Cornbelt region. We have bioreactor. We have treatment wetlands. We also have conservational drainage and grass waterway. The reason that we're mostly interested in bioreactor is because, first of all, in the Midwestern region, we have extensive drain tile system. Oh, there we go. So that is the cockroach in the bioreactor. It was quite nice. Well, anyways, yeah, that is the maps from nutrient reduction region. So you can see Minnesota River Basin here is quite a prioritized zone for any type of treatment. Oh yeah, that's what I'm missing. So the nitrification overview, it is one of the most important processes in the nitrogen cycle. It returns nitrogen into the atmosphere. So it basically involves 5 steps: nitrate, nitrite, nitric oxide, nitrous oxide, and dinitrogen gas. The reason I was highlighting nitrous oxide is because It is one of the greenhouse gases, and it is 300 times more effective than carbon dioxide. It has a relatively long life cycle too, so a tiny amount can have a relatively huge impact on global climate change. So this one is bioreactor, that one is treatment wetland, down there is a grass waterway. It's actually saturated buffer. So we stopped here. The reason why we chose bioreactor, first of all, I talked about the extensive drainage system, relatively consistent nitrate concentration over a drainage season at a given site. So that ensures that we have enough food for the microbes, and they will have a, oh, okay, thanks. All right, so we provide food constantly for them, and they'll have relatively stable environment to grow. The other reason is subsurface flow. Treatment wetlands and saturated buffers, they're great for surface flow treatment. But for Minnesota and the Cornbelt region, we drain the water out of the field using tile drains. So bioreactor is actually underground at the end of all the drain tile outlets. So it is a great way to deal with the subsurface flow. The third one is biostimulation. How it works is we simply just provide food for the microbes. And because denitrification is naturally occurring in the soils, so we do not have to introduce any invasive species to the site. So it's environmental friendly, and it's quite simple. It also has a really high reduction rate, which we'll talk about in my result. And the space is another issue because we're dealing with agricultural field, and we talk to a lot of farmers. They get kind of angry when you're trying to take away their farmland to put in those treatment strategies. When we sell bioreactor, we would tell them it's underground, it's at the drain tile outlets, it's not going to take up all your surface soil, and you actually can grow crops on top of bioreactors. It's not going to be affected. So because the field study difficulty, we started the lab scale study. And our design is kind of different than a lot of lab studies. Most people would choose to use a vertical flow, so you do not have to deal with water bypassing and water mixing problems. But here we chose to do a horizontal flow, which is more, which is closer to the field condition. So we have two bioreactors, 1 and 2, and each of them have 3 chambers. I'm going to draw it here. So the design that we had to achieve a desired length and width ratio. So this is a side view of it. So we actually put 2 baffles into the bioreactor, each chamber. One goes this way, one goes the other way. So our water flow direction would come in from here, travel all the way to the end, get pushed up, go all the way to the other end, and get pushed up, and then come out from this end. And you know that denitrification requires a strict anaerobic condition because nitrate is right below the oxygen energy level. So when there is no oxygen available to the microbes, they would automatically go to nitrate. That's why we took soil from the site and capped this whole bioreactor with soil on top. And when you run water through it, you have a relatively saturated soil, and that creates the anaerobic condition. So the variables we're looking at, first of all, is alternative carbon sources. It's one of the controlling factors for denitrification rate. And we tested 3 different carbon sources. First of all, is woodchip. It's a mix of hardwood. It is the most commonly used type of carbon source in this kind of bioreactor. Second one is the biochar. It's becoming a new favorite thing for a lot of soil scientists. Research has proven that biochar is a great soil amendment. It retains nitrogen. So we want to see if it has any effect in nitrate and phosphorus removal. Our idea was to create cheap and accessible material. So instead of buying biochar or try to create the oven and the whole process in the lab, we did it on site, and we call it caramelized wood chips. And the last one is corn cobs. For a huge corn production state, we have a lot of corn cob available. And the nice thing to know about corn cobs is that it has a softer core compared to other two material. So a lot of times the microbes actually prefer a softer core. And then the other limiting factor we were looking at was residence time. Residence time defines how long the pollutant can stay in the bioreactor before it comes out. And we tested 24 hour and 8 hours. 24 is more like an ideal. 24 hours is more like an ideal situation. It's more like measuring the potential. And 8 hour is a lot more close. 8 hour is closer to the actual field condition. And other controlling factors, we have nitrate concentration. We kept it around 21 ppm. And temperature, we used room temperature, but in Minnesota, the temperature dropped like below 30 all the time in the winter. So it kind of got cold in the lab because the thermostats got turned off. And another thing is oxygen availability, which we use the soil to create the anaerobic condition. And nutrient removal, we tested two nutrients, nitrate and phosphorus. Nitrate, it's just NO3, and phosphorus, we actually measured orthophosphate because there are two phases of phosphorus. Usually phosphorus get attached to soil particles, but it's not commonly seen when you have a tile drain system. Orthophosphate is the most commonly seen dissolved phosphorus form. And also we measure nitrous oxide production. We're using the chamber technique. And we run bromide test through those bioreactors so to compare with the calculated residence time. So at a 24-hour residence time, our flow rate was about 50 mils per minute, and at an 8-hour residence time, our flow rate was about 150. So here are the results that I got from the lab study, 24-hour. Up here, the light dots, it is the input nitrate concentration. It kept quite consistent. And the blue line here is actually the temperature. I've been keeping track of every day. This is a daily time step. And this one, the red one, is biochar. The green one is corn cobs, and the blue one is woodchip. We can see that woodchip had a great reduction potential. I'm actually going to switch to the next graph because this is a percentage. So the reduction percentage for nitrate from woodchip stays well above 95%. And from corn cobs, it's the most stable one here. It's around 90. The interesting about biochar is it varies a lot, and we were wondering why. So we did a correlation study with the temperature using statistical software. And you can see that there's some sort of correlation between those two, like the change in the curves. So actually, we got a result about 0.5. For woodchip, the correlation was about 0.2. For corn cobs, it's 0.05. So compared to those two, and considering it's a biological process, 0.5 is actually pretty highly correlated. And the phosphorus reduction is pretty promising. the percentage, this blue line is at 50%. Everything stays well above 50%. And it's worth mentioning that biochar stays really high after the first month. So it has a great reduction potential at a 24-hour residence time. And the 24-hour study for the nitrous oxide, we can see that biochar is not that desirable. It has a lot more nitrous oxide emission, and it actually kind of matches with other results from other studies because you change the carbon-nitrogen ratio. So sometimes they will emit more nitrous oxide at the earlier stages. And for 8 hours, it got a lot messier. Still, the corn cob is relatively low over here. So we kind of know that although we switch residence time, corn cob is relatively consistent compared to the other two. That's a percentage. And biochar we do not recommend for a really high residence time because it's only about 20% reduction. So phosphorus reduction for 8-hour residence time is relatively consistent among the three. They have the same trend. That's a percentage. It's everything stays almost above 50%. And the nitrous oxide emission is the same trend between 24 hours and 8 hours. So just quick summary and last couple comments on the media. Woodchip is great for nitrate removal. Biochar, it is great for orthophosphate removal depending on the residence time. But nitrous oxide is one thing to be concerned. However, the thing is that when you have a relatively lower nitrate reduction rate, it's actually used up slower than woodchip. So we would predict that if you put biochar in the bioreactor, it might last longer than the other two. Corn cob had really great environmental resilience regarding temperature change and residence time change. It always remove a lot of nitrate and phosphorus. So questions, comments?
Okay, so any question? Yeah. So please.
You mentioned that the idea is to install these in the field with the farmers, and you have problems, and you have to do this in the lab.
Yeah.
But you did this under temperature control condition, about 21 degrees, and you notice an interaction or an effect of temperature.
Yeah, we do.
Given that you have small changes in temperature.
Yeah.
It's true.
Yeah,
you have about 2 degrees difference in temperature.
Mm-hmm.
But in the field, you are going to find under frost conditions in the winter and very hot conditions in the summer.
Yeah.
So what is your comment about that?
In the summer in Minnesota, we see a lot of nitrate reduction, of course, because the temperature is relatively ideal. But in the winter, study has shown that there's a little bit of denitrification going on because they do have bioreactor in the field at the other department. They only tested woodchip. But I say that there's still a potential for it for the cold weather denitrification to go on. And based on this data, we can predict that if you have biochar in the field bioreactor, it might respond to the temperature more sensitively. We would recommend corn cobs, but in the winter, it's not a really high flow, so it stays in the it could stay in the bioreactor a lot longer for actual removal.
Okay, perfect. So thank you very much once again. So let's thank Mrs. Luzan. So we are giving you a certificate for recognition for the presentation. Thank you.
Thank you very
I'm
much.
sorry for the
And this for memory.
logistic problems.